morning, class.
Good morning.
So, have you ever seen movies of the series like CID? Have you seen?
Yes.
Okay, so what have you seen there? There are the crime scene and the forensic team will come and they will collect the blood sample,
Yeah.
thumb sample or the hair follicles. So what they do? What is the basis of all this? So we are going to study today about DNA fingerprinting. DNA fingerprinting is also known as DNA profiling. So basically it is the process of analyzing the DNA sequences there is having similarity or the differences in the DNA sequences of two individuals. DNA fingerprinting was given by Alfred Jeffrey sir, and he was the first person who done this process. So we will study about the DNA fingerprinting. Here we can see it is the DNA. Suppose it is my DNA. So in this DNA, there is 99.1% of the similar DNA. That means my DNA of 99.1% and your DNA of 99.1% is similar. But there is 0.1% of the DNA which is known as the satellite DNA is unique for everyone. I am having the different satellite DNA and you all is having different satellite DNA. There are some properties of the satellite DNA. First is the satellite DNA is the non-coding part of the DNA. That means it do not code for any protein. The normal function of the DNA is to do replication and then the transcription and translation to prepare protein. But the satellite DNA do not code for any DNA. That's why it is also known as the junk DNA. Second is the polymorphism. Satellite DNA is responsible for the variations among the person, that I am looking different and you all are looking different. So it shows the polymorphism among the individuals. Third is the junk DNA, as I have mentioned. It is not making any protein, that's why it is also known as the junk DNA. Fourth is the repeats of the short sequences. So in all the satellite DNA, there are the repeats of short sequences. Suppose like ATTG, ATTG, ATTG. It can be 3 in number, 4 in number, 5 in number. That's why these are known as the VNTR, variable numbers of the tandem repeats. That means it can be 3, 4, 5 or more. And this sequence also can be different. I am just providing here the example. So this is about the satellite DNA. But how we will do the process of the DNA fingerprinting? Let's look. So the technique of the DNA fingerprinting. First thing is we will isolate the DNA. The process is DNA isolation. How we will do? So we will take a test tube. In it there is a sucrose solution. We will put our DNA into it. Okay. Now tell me that the heavy portion of the DNA will go where? Will go to
down.
the bottom of the test tube. And the light DNA will come into the
Surface
of the DNA test tube. And how it can be achieved? By the density gradient centrifugation. Centrifugation means we will spin it into the machine and it will make the light DNA and the heavy DNA. Heavy DNA is the similar DNA and the light DNA is the satellite DNA. We will extract it from here. So DNA isolation is done. After the isolation of the DNA, we will amplify the DNA. Why we need amplification? Amplification means we will make the multiple copies of the DNA, okay, and we will keep it, as there are chances of the damage of the DNA. That's why we make multiple copies. How we make multiple copies? Through PCR, polymerase chain reaction. So we make multiple copies of our DNA. Third process is DNA digestion. What is DNA digestion? As we know, we have a long length of the DNA. So how we will analyze this much of length? So we will make a small fragments of the DNA. Whole DNA is break down into small fragments through restriction endonuclease. Restriction endonuclease is used for the DNA digestion. As we have made the small fragments of the DNA, we will move towards the DNA separation. How we will separate the DNA? We got multiple segments of the DNA. Now we have to separate according to the size of the DNA. So what we will do? We will separate the DNA by the gel electrophoresis process. Gel electrophoresis is the process where DNA move from negative to positive side according to the size of the band. Just like this. This is the it look like. So here we can see the small band will move towards the positive side and so on. Here we can see the bands of the DNA, but we cannot see it through our eyes. So what we will do? We will do a different process. It is known as the Southern blotting. Southern blotting is a process where we transfer this bands into the membrane, and this membrane can be of nylon or nitrocellulose. We will transfer it into this. We have transferred it into the membrane. Southern blotting is done. After transfer it into the membrane, we will do hybridization. To understand hybridization, first we have to know what is probe. So probe are lab synthesized small fragments of the DNA that are single stranded. Second, these are radioactive. So what we will do with the probe? The probe will go and bind with the DNA single stranded VNTRs that are present here. It will look like this. It will attach to the complementary nucleotide sequences. Here you can see. Now we can see this, and we will take a X-ray film of the autoradiographic exposure. Okay? And now then we can see the dark or the light bands in the X-ray film. These probes we have used is of the another person we are matching our DNA, and the DNA, this DNA is of the first person we have taken from the crime site or the first person. So what is the application of the DNA fingerprinting? First, it is used for the crime investigations, where the blood sample or the hair follicle or the thumbprint is found and we match with the suspects of the that investigation. Second is for the paternity test. So there are some disputes. To settle these disputes, we use paternity test. Third is to know the close relatives of in the population. And these are the process of the DNA fingerprinting. I hope you like it. Thank you so much.